presentation. So, as has been alluded to throughout this workshop, and it's fitting that this is coming towards the end, that one of the issues we're trying to grasp with throughout this, these discussions and talks is that timbre is a dimension that is, as we hopefully everybody agrees, hard to define, not trivial to quantify, and unlike the other attributes of sound, when you ask the question, what are you really listening for to tell different instruments from each other, it's not a trivial question to answer. The other point that I wanted to bring up, just that since it came up both in Jeffrey's talk but also in some of the talks yesterday, is that it does impact both the identification, the recognition. You can tell a violin from a saxophone, but it also has to do with how we judge sounds in their context, specifically in the when we talk about musical timbre. The most important thing that I wanted to emphasize is we don't want to lose sight of the fact that when we talk about timbre, it is a perceptual dimension. It is something that reflects how we perceive it in our brain. It's not an acoustic attribute that you go and measure in the sound itself. And so, looking at the numerous studies that have tried to actually boil down the space of timbre into acoustic dimensions, everybody agrees that there is a wide range of spectral and temporal attributes that correlate with timbre in one way or another. And as we just heard in this previous talk, you can boil it down to 5 dimensions that probably span a subset of these acoustic features and would be represented in this modulation spectrum space that somehow captures some of these. But the key important thing that I wanted to, not lose sight of is that it is a time and frequency combined space. And so when we ask the question, what do we listen for when we tell a saxophone from a violin, we can take inspiration from some of the physiological investigations about how sounds are represented in the auditory system, particularly at the level of auditory cortex, in that we know that auditory cortex neurons are involved in extracting both spectral and temporal information about complex sounds. And then the natural question that arises is, could this be a possible neural underpinning of timbre? So this is a simplified diagram of the mammalian auditory pathway. When sounds come to our ears, they travel through these different stages where a lot of acoustic features get extracted. What is of interest to us here is the level of auditory cortex, because based on what we know about the representation of these different stages, auditory cortex seems to play an important role in representing sort of more complex sounds, speech, natural sounds, and so musical sounds would also fit in that category. And I'm going to tell you a little bit about why auditory cortex is a site of interest here. So typically the way we would study these different nuclei in the auditory pathway is using this sort of systems theory approach. Think of it as a black box. You play a sound to the ear, you record the neural response, and you think of this as a system, and then you try to characterize what transformation happens from this input to give me that output. So most of the results I'm going to be showing you here today were recorded in ferrets. This is a picture of the setup. But similar results were obtained in other mammalian systems, the cat, in rats and monkeys. And so, as I said, the idea is this sort of black box approach. You play a sound, you record a response, and you try to guess what happened in between. So, because we are dealing with a complex system, namely the brain, it is not a trivial mapping to go from input to output. And so, throughout a number of investigations, the linearized approach has been favored. Namely, you assume that this transformation is a linear system. And so you use this sort of linear convolution-based approach, namely that the stimulus gets convolved with some transfer function of the system, and that gives you the output. Now, the reason we are putting this equation in time and frequency is because this is specific to auditory cortex. So when you go to the level, so when you record in earlier stages of the pathway, you can apply the same idea in a sort of typical convolution way. Sound comes in as a time dimension. You can convolve it with a filter. That's certainly true for the cochlea, and you get a response. When you get to higher stages, particularly even actually starting from the midbrain, but certainly true in the cortex, cortical neurons don't like to respond to boring sounds. So if you give them a pure tone, they'll give you an onset response and then they'll shut down. Boring, sounds, meaning a pure tone, a stationary sound, just a white noise that goes psh. The neuron gives you an onset response and then it shuts down. There is nothing interesting about it dynamically. The sound is the same and does not change. And so beyond that onset response, auditory cortex neurons don't like to respond to that. In contrast, and thanks to Tafta for introducing the ripples, I don't have to play them. In contrast, when you give the auditory cortex something more modulated, that changes, that is more rich, they like to respond to these sounds. I think just to contrast these ripples, so this, as Tafta introduced, I don't have to do that again. This is a spectrogram of many of these ripples. So these are basically noises that have been modulated in time and in frequency, and you can vary their modulations in time to make it denser or coarser, and similarly in frequency you can make it denser or coarser in frequency. So this is the analogous idea of a pure tone, but in two-dimensional space. And as I said, the reason for doing that is because auditory cortex does not like pure tones, because pure tones are stationary, they have a stationary envelope over time, while a sound like this has more dynamics and changes, is more interesting. So as you vary these modulations, the perceptual quality of the sound changes, and it feels sort of, the information either over time or over frequency gets represented in a different way. So I'll just play a few examples of these ripples. So you see this nice contrast in the temporal dynamics, and similarly between these two ripples that have different spectral modulations. Okay, so this is just varying these modulations. So mathematically, it is a very simple sort of sinusoidal type sound. This is representing basically the carrier that sits on top of a white noise sound. But because we're using these pure sinusoids to drive auditory cortex neurons, we can ask similar questions like we would do when we derive the receptive field, the transfer function of a system. You play a sinusoid, you record the response, and that tells you whether that system likes that sinusoid or not. So here we do that the same way, but we do it in two-dimensional space. So we play this ripple, we record the response, we play that ripple, we record the response. So this is the kind of response you would get. This is an example of a neuroresponse from one neuron. Each dot here is a spike that neuron responds to. And this is varying the modulations in time, so 4 hertz, 8 hertz, 12 hertz, etc. And you see that the neuron, for example, when you play an 8 hertz ripple, gives you, so this is just each line here is one stimulus, and you repeat the same 8 hertz many times because neurons are not deterministic systems, and there is a probabilistic nature to the way they respond, so you would typically present the same stimulus many times. But so when you're playing this 8 hertz ripple, you see that the neuron gives you a spike at a rate that follows the 8 hertz. This neuron likes an 8 hertz dynamic, and so it gives you a response at a rate that follows the 8 hertz. By the time you go to very high modulations, this neuron cannot follow. This is way too fast, and so that's an indication that this neuron does not like 28 hertz. So when you do that over all the spectral temporal space, you play one ripple at a time, then you get basically an estimate of how well the neuron responds to each of these ripples. So each dot here is the response to one of the ripples, and you do that many times. Then when you take the inverse 2D Fourier transform, you get a time frequency representation that we call the receptive field, the spectral temporal receptive field of the neuron. Now the way you read this is basically, so the colors are not the typical ones you see in a picture because of the projector, but so this is time and frequency. So in red, it shows you the frequencies that the neuron likes to respond to. in this sort of yellowish color is sort of a mid-level response that excites the neuron. This blue, sort of greenish blue, response here is what we call inhibition. These are patterns that would shut down the neuron, and this baseline white, light blue thing is sort of a baseline response. The neuron doesn't care about these kinds of patterns. So this is a picture that shows you 6 different neurons recorded in an awake ferret. So what you basically, the important things to note is that each of these neurons likes to respond to different patterns in sound. So you see this neuron likes to respond to 500 hertz, around 500 hertz. This one likes to respond to broadband sounds that span a lot of frequency. This is a neuron that's rather slow temporally, so it likes sounds that go on and off within 100 milliseconds. This one is a sort of short one that likes sounds that go on and off within 50 milliseconds. And so we're looking at 6 neurons. We have 1 billion of those in our auditory cortex. And so you imagine now when you take the variety of neurons over many studies that people looked at these cortical spectrotemporal receptive fields, it turns out that the auditory cortex represents sounds through this pattern of these receptive fields. Think of them as sort of different prisms. So sound comes in, and you have 1 billion of these, each tuned to a little detail in the sound. So the sound comes in, it gets projected through this array of these billions of cortical neurons, and each one of them tells it something about what the sound has. It tells it something about the spectral details in this particular region, how sound is moving, whether it's moving up or down, these modulations, the sweeps in the signal, the dynamics over time, the temporal modulations, whether the sound is broadband or narrowband. So it's really one of the important things that I think you get inspired by thinking about this neurophysiological perspective about how the brain processes sound, is that we try to boil down features into the minimal representative descriptors. The brain does the opposite of that. It wants to map sound onto a very high dimensional space. It's not going into, do I have just these few representations? It's really taking the signal and spreading it onto a very high dimensional space that represents time, frequency, temporal modulation, spectral modulation. So this would be the analogous point that you were commenting on. So instead of taking just a 2D Fourier transform of that, it's basically taking that, convolving it through each of these filters, so now it knows something about the modulations in time and frequency, but also where they are in the time-frequency domain. So this idea of the brain mapping the sound onto a high-dimensional space is very interesting because it not only emphasizes the important role of modulations, that somehow modulations are a very important aspect of how we perceive sounds, but it also gives us a sense that this high-dimensional representation that one has to keep in mind is very redundant. There is nothing that says that the features represented in this part of the space are different than the other ones. It's a highly redundant representation. Nonetheless, what we learn from the cortical representation is that that's an important factor in how we process sounds. So then the natural question comes, well, how well does this cortical processing capture dimensions of timbre? So we ask this question directly. We basically take a musical note that we process through an array of these receptive fields that get mapped onto this multidimensional space, as I said, and then we just ask the question, well, how good is this space in telling us something about musical timbre? So the way we did that is we basically took the RWC database that was mentioned earlier. That's 11 instruments, full pitch range for each instrument. So that gives us about 2,000 notes per instrument from a variety of playing styles and manufacturers. We took an array of these receptive fields that were recorded physiologically from 15 different animals through different studies. And then we mapped each of these notes through these 1,000 receptive fields. That gives us a very high dimensional space that we then ask, can we discriminate different instruments in this space? Now comes the limitation of machine learning. We're dealing with a very high dimensional space. How are we going to do this? So we reduce this space to 400 and 20. We chose that because it gives us the most variance in this space. We also had to add a Gaussian kernel warping. It's basically a nonlinearity that we add on this space. And so when we ask the question, how well can we identify musical instruments, we're able to do that with an 87% accuracy, which was quite encouraging. We then noted that we are using these neurophysiologically recorded receptive fields that tend to be biased. So when you do these experiments, you stick an electrode in this animal's brain, you're recording from a specific region, you're not sampling the full billion neurons that these cortical, that the cortical area has. So we instead used simulated cortical receptive fields that allow us to span the space more completely, because the physiologically recorded ones tend to undersample the space. So we increased the number of receptive fields to 30,000. So instead of these 1,000, we use the simulated ones that we brought up to 30,000, but then we had to reduce once we do this mapping. And then, lo and behold, the ability to identify instruments goes way high, which basically we are almost at a perfect ability to discriminate instruments in this database. Now, the next question we asked is, and just as a side note, when we apply sort of a pre-cortical level representation, we are able to achieve about 80% recognition accuracy. Now, the important point that I don't want to finish before getting to is that, so as was mentioned in Jeff Ross' talk, is that a lot of times we choose some features to do recognition, and then we choose some maybe different features to try to map this space of distances between musical instruments. So we wanted to ask the same question. Using these same features, now we know that we are almost 100%. That means when you map the sound through these cortical features, you can tell a violin from a cello from a saxophone. But do they map onto a space that corresponds to the perceptual space that we know humans judge the distances between these instruments? So we basically replicated the classic McAdams perceptual judgment study, basically doing a similarity. So this is work that was done in collaboration with Daniel Kresnitsia. So basically getting the dissimilarity between pairs of instruments at 3 different pitches. This is showing the mean for each of the 3 pitches, as well as the standard deviation. So we average across the 3 pitches because we are focusing on the timbre aspect of the question. And then we ask, well, how well does the cortical processing? So if we take this cortical space and now measure the distance between the representation of pairs of instruments, how well does it give us this human dissimilarity matrix? And so the results basically give us a matching of a 0.94, which is, again, very interesting because it says that not only does this modulation space capture maps the instruments into different parts of the space, but the distance where it maps them is quite well correlated with how humans represent that mapping between those spaces. So just as some side notes, when we use the physiologically recorded SCRFs, we get a lower accuracy of 0.73. Again, the reason here is these are biased by where you're recording in the animal's brain. We don't have a full mapping of the modulation space through these recorded SCRFs, and so the simulated ones give us a full range or full representation. One of the interesting things that we find out is that when we just use the space without this nonlinear warping, we don't do quite as well. So the correlation with the human similarity matrix is 0.6, which suggests that the linearized approach to how the brain represents sound is missing the nonlinearities that the brain has, which brings about a lot of the power in the perceptual representation of these complex sounds. So some final analyses that we did on this data. So we basically took our feature space and we wanted to know what parts of these cortical representations are the most important. So we asked whether the auditory spectrum features can also replicate these similarity, the human similarity matrix, and that gives us a correlation of about 0.74. These two are actually the most interesting ones. So in one we asked how well do the global modulation features, which are exactly what we get from the global representation of the basically a 2D Fourier transform of the spectrogram. So this don't have the details of where these modulations are. They just tell you that these modulations exist in the sound, and they give us a correlation of about 0.7. That does not mean that they may not be able to distinguish the instruments, but in terms of mapping them into the right perceptual space, they seem to be limited. The other very interesting one is this one. So in this one, we basically just took the temporal modulations alone and the spectral modulations alone, and we concatenated them. So what we took away are these oriented ripples. We just took the horizontal ones and the vertical ones. So the modulation information is still there, but there is no information about the tilts, the sweeps, the FM sweeps in the signal, and that actually hurts how well we can match the perceptual space. So all of this together basically gives us evidence that the representation at the level of primary auditory cortex could potentially underlie how we perceive musical timbre. It does confirm this importance of this multidimensional nature of the timbre percept. It is very important to keep in mind that it is a joint time-frequency space. It is not time and frequency separate, but time with frequency together. That the representation of the sound identity is not really just reflected in a simple set. It's not a laundry list of features that you want to add together to get to the representation. At least when you ask the question perceptually, what is the meaning of timbre in that domain? If it's sort of a specific application, I think that's fine. But when you ask the question, well, how do we actually perceive musical timbre, then it is really a complex perceptual space. And the other point that is we should not lose sight of is that even though these cortical representations that I showed you are able to give us almost 100% recognition of instruments, we are able to do that because we introduce some nonlinearity that we don't really know how the brain does this. We are doing our own machine learning based nonlinearity. How the brain actually maps this cortical space with its own nonlinearities is unknown. We know something about some of the nonlinear processes that go on in the brain, the divisive normalization, synaptic depression, the way neurons respond. We are linearizing their response through these receptive fields, but they're not actually a linear process. The way they respond to onsets is not the same as they respond to steady state, and there is a lot to be investigated as far as how really the brain maps sounds fully taking into account these nonlinear processes. So finally, I just wanted to point out that one of the things that I think is interesting in this STRF-based approach is that it's not, I don't view it as yet another acoustic descriptor, just in reference to Jeff West's talk. I think it tells us something very important about this concept of timbre, that there is the space of modulation is capturing not just the acoustic attributes of the sound for globally, but it gives us a lot of details about how the sound evolves, and it seems that that's important, at least the brain thinks it's important in terms of how it represents the sound, and it seems to also matter how we perceive, how these sounds get perceived perceptually. And so we may disagree on how to implement these modulations. We may have different approaches. The approach, for example, that Stéphane Mallat is using is an approximation of this. This may not be the approach. STRF is an approximation, but I think the concept of modulation is very critical. It is at the core of how one would like to think about the representation of complex sound, especially when it comes to musical timbre. Different implementations may disagree, but I think if they miss sight of how to represent these modulations, they're probably going to be limited in what they can do, at least when it comes to addressing the question of musical timbre. So I'm just going to stop here, acknowledge the funding agencies, and take any questions. Thank you.
I'm curious about the steps of the marginality reduction and, for example, this kernel walk-in, in the sense, or whether it preserves the acoustic interpretability of the model. So say I would be interested in figuring out whether the onset or the offset transient of a sound is critical for identification. How would I go about to identify this in the model?
I think I agree with you. That's where I said we sort of we follow the brain's lead and go to this high-dimensional space, and then we're stuck there. We need to do something with that space, and so we go the machine learning approach and say reduce dimensionality. And so in that process, we do lose sight of these kinds of questions. We can do the regression and go back and say, if I change this, what does it do there? But yes, that is definitely something that we do in order to answer the question, can I recognize the violin through this space? But naturally, the brain has its own nonlinear processing, and that's something that is still unknown and yet to be investigated. So,
so thank you very much. This is excellent, and I really like the biological justification. So my question was related to the fact that in your experiment you first take the impulse response you get on the ferret. Then after you move to 30,000 simulated receptive fields, and so you highly increase the dimensionality. And I was wondering, so you somehow discretize the modulation and rate space, and why 30,000? And would you have even better result if you move to 1 million receptive fields? So what is this notion of discretization of rate and modulation? So is it the case in the neurons? Because in the figures it seems continuous. So can you comment on moving from continuous to discrete?
Yeah, so this image, I didn't pause on it. So what we did here is we took the physiological neurons and we looked at where their best energy is. So these are so when you take this one neuron that you record, where is its best energy? So this what happens to be at 4 Hertz and a quarter. By the way, just as a side note, I'm using spectral modulations in cycles per octave. I think Takata presented them as cycle per kilohertz. We represent the frequency axis in log, so it's just a minor mapping. So this one neuron happens to be at 4 Hertz and a quarter cycles per octave. So we did that for different neurons, just looking at their peak energy. They have other stuff, but we looked at their peak energy, and you see how big the dot is where most recorded STRFs were. So we have a lot of them here, only a few there. So what we did is we put more simulated neurons to make an equal tiling. But I do agree with you. This is an approximation to this space because we're not putting anything here. The assumption is when you build this simulated receptive field, it sort of has a little bump around there. The brain, as I said, at least auditory cortex by many estimates has about 1 billion of these. There is nothing that says we should not maybe do better with 1 billion. Maybe for this task that we were working with, that was 30,000 was enough to give us, close to 100%. Maybe when you start asking more difficult questions, if we now go into sort of these are individual notes. If we go to orchestral music, maybe it's going to be we need all of them to help us. But this is just an artificial tiling of that space. There is nothing, scientifically motivated about how we went about.
Yeah. And just a short side question, because in the previous presentation we had linear frequency scale, which I think is motivated by the fact that harmonic signal is just a peak in the 2D Fourier transform. And here it's based on biological evidence that show that it is logarithmic. So, what's best?
I don't think there is a best. I mean, they both map to each other. The difference is when you have a harmonic stack, you're not going to see it as one dot in this space. You're going to see it spread. You see one peak at one cycle per. I'm sorry?
It's not sparse anymore.
But I don't think there is a purpose of sparsity. If anything, as I said, it seems that the brain wants redundancy. I don't think it's trying to go sparse. So I don't think there's anything wrong. I think sparse will like it because, exactly for that question, once you have this high-dimensional space, then you start swimming in dimensionality and you need to do shortcuts to cut it down, but the brain doesn't seem to have that problem.
So the conclusion is don't be sparse.
I think redundancy seems to be our friend here, if anything.
Okay.
The question, how many receptive fields do we need, leads to another question. How fixed or born are these structures, or are they also the result of a learning process?
So that gets us to another dimension of this receptive field. So these receptive fields that I've shown you were recorded in passive animals. So these are animals that are sitting there, you play them a sound, they're not doing anything in particular. When you engage an animal in a task, it's paying attention to a sound, it's there is a sound that impacts its, it's going to get water if it responds to a certain sound pattern, and these receptive fields start changing. These filters now move their tune-in, so a neuron that what might have been here, if there is a pure tone of interest here, the neuron might grow a little bit more. And then when the animal is done with the task, the neuron goes back to its state. So these are what we call sort of the passive state of the brain. The animal may be dozing off or just sitting there bored while you're playing these ripples. But the fact of the matter is, in everyday life, as I'm talking to you, chances are your cortical neurons are now sort of modulating their properties a little bit. They don't shift completely. It's not like this neuron is going to go all the way here. They just sort of locally change to help you focus on sounds of interest or process sounds differently. So if you're trying to pay attention to my voice, there's probably a little bit of tuning towards some of the acoustic properties. So there is another complicated layer to this whole representation that we're not capturing at all here, because these are neurons that were reported in another context. We just took neurons that were reported in other studies and asked the question, how well do they represent musical timbre? It would be definitely interesting to actually record from animals that are engaged in a task that may use musical instruments or musical notes. That would be definitely very fascinating. I don't know if anybody is actually doing that. So, yeah.
I'm still curious about the multidimensional space and high dimensionality of the space. I was thinking about how many dimensions you have, and the question to me was exactly that, sparsity. I was thinking about the curse of dimensionality and how, you start to increase the number of dimensions, everything starts to become really far apart, and that's not what you want. What you want is, like you said, you want things to overlap and...
Well, not overlap. I think it is using, it seems to me the way the brain solves this problem is when you look in the acoustic waveform, everything is on top of each other when you look at a sound waveform. So what the brain seems to be doing is it's, takes the acoustic waveform and spreads it onto a high-dimensional space, so everything is well represented within itself. How it samples that space is where I think we now are still limited in our understanding. So it has all the information, so where does it go to pick where is the most relevant? And that's where we end up doing dimensionality reduction and all these other things. And I think that's where a lot of neuroscience needs to answer is, now you've represented, you have this rich, high-dimensional, redundant representation, so how does the brain actually take that and form a percept? I don't think we know the answer to that. And that's where we start taking shortcuts about, sort of Gaussian kernel warping and...
But the processing, you're saying it's isolated. One neuron is responding to this very particular...
Each neuron seems to have its own characteristics that may overlap with a lot of other neurons, but yes, when you look at 100 neurons, you will not see two that are exactly the same. So it may be tiling this space really finely, but it's using 1 billion neurons to do that. Does it use all of them? I don't think we know the answer to that.
It's not at all the neural network, like 100 billion connections.
It is. There are connections. So these are not treating these in parallel. These neurons are connected with each other. I don't think anybody knows how to read out that.
Last question.
But so in the dimensionality reduction you did, so you went down to 420, you call them features. What do you mean by, what are these features actually? Like, did you find, and I don't know if that's actually correct, what I'm about to say, did you find regions of neurons that, so because you present, let's say, all the space with the neurons, right, and the logarithmic distance in the modulations, but then I imagine when the reduction, the dimensionality reduction comes, so it finds that there are particular groups of neurons, maybe group is a better word than region, that keeps the variability going.
So it's not really a sampling, because if it was a sampling, we can answer the question that I asked better, which neurons are more active, like when you have an onset. So it's really, you take all of the 30,000 or 100,000 representation, and then you do a PCA. We did different things. You really take all of it and you transform it into a number of dimensions that capture the variance.
Did you look at the PCA results?
Yes.
And
So, I mean, you can infer certain things, but I think we know that that's not how the brain, you know, it must be reduced in dimensionality at some point because it comes back to a percept, to a fixed percept. But I don't know of any study that has informed us about how to go about doing that. So, but when you do PCA, yeah, you see some interesting things. You see, you know, strings tend to be more represented in certain dimensions, and you know, you can infer things. But I think that's, you know, you get some of the trivial observations that you would expect.
Thank you very much.